for the kind introduction. It's a pleasure to be here. I really enjoyed yesterday, and I'm looking forward to the rest of today after the next hour. So let me begin and try to tell you what a little bit of history, and then mostly some quite contemporary work from my lab. And I want to begin by a disclaimer and say that I think talks are for entertainment. That is, you're supposed to be, your attention is supposed to be grabbed by what I have to say. And that's not necessarily the entire truth. They are often mutually exclusive. So you have to read papers if you want to see the whole story. And so this story began more or less with the beginning of the Earth 4+ billion years ago, with this inexorable light-dark cycle that really governed the evolution of life on Earth. This 24-hour waning and waxing of illumination, along with, in most of the Earth, changes in temperature, really was the original Zeitgeber, the most important environmental stimulus that life emerged. The echo here is quite prominent. I don't know if the previous speakers have experienced that. Let's see if that's a little better. So I need to say really at the outset that circadian rhythms are so important that they've evolved multiple times in evolution. And we infer this because the clock which runs in bacteria, in cyanobacteria most notably, a fantastic, beautiful clock, has absolutely no molecular connection whatsoever with the clock that operates in animals and probably also in plants. So there's really no way to interpret that other than independent origins of the clocks in these different kingdoms. And let me just put everybody on the same page by saying a word or two about circadian rhythm properties. So they continue in constant darkness. In other words, in the absence of the light-dark cycle, the clock keeps on ticking, and it does so with not precisely 24-hour period. In other words, our clock is about 24 hours and 15 minutes. Fruit flies have a clock that's just a tiny bit shorter than 24 hours. So circadian circadia, about a day. And yet it is the light-dark cycle that entrains the clock every day to precisely 24 hours, because that light-dark cycle can reset our slightly slow clock back so that we're on the same schedule every day. And I should just say that I'm a great believer in accidents of life, as I was explaining to Giro yesterday in the cab. The fact that I work on circadian rhythms is 100% due to the fact that I came to Brandeis by accident. And then I teamed up with my friend and colleague, Jeff Hall, for 20 years. Jeff had been trained in Seymour Benzer's lab, and Jeff knew about circadian rhythms, and I had never heard of such a thing. And if had I gone to another university, I'm sure I would be working on something else, and none of this would have taken place. And so, speaking of Seymour, the field owes a debt, an incredible debt of gratitude to this paper and these two individuals, namely Ron Konopka and Seymour Benzer, for their fantastic paper published in 1971, in which they identified 3 alleles of the period gene: a fast allele, a slow allele, and an arrhythmic allele, all single nucleotide changes in one gene, remarkable, faster, slower, and no clock whatsoever. And proposed, of course, that this gene was central in some way to the operating of the clock. I should say, for those of you who chase after high-profile publications, this paper in the first decade after its publication had 10 citations. So its longevity and importance in the end, and not the immediate impact necessarily of a paper that really mattered. So we became famous. I let's say, received that very gracious introduction because of three reasons, three consequences of our early work on circadian rhythms. First of all, it turned out that our fruit fly work, which would have died in obscurity if it hadn't been for the fact that the people coming after us discovered that exactly the same mechanism, the same feedback loop operated in mammals. And so, so the small feedback loop, the transcription translation feedback loop that operates in flies, was inherited from a common ancestor, and mammals, including humans, inherited exactly the same system. Not only the same system, but the same genes operate there. They're really orthologs. 3 of the 4 genes are complete orthologs in the 2 systems. So that's the first reason. The second reason is because this clock does not just operate in a few places, for instance, the brain, the suprachiasmatic nucleus in mammals, for example, but it operates all over animals in all or nearly all tissues in an animal. So it's governing the liver, heart, lung, spleen, skin in a semi-cell autonomous or tissue autonomous fashion. And of course, that fact, coupled with the third principle, that is that this little feedback loop is an umbrella over almost all of physiology, not only because of the fact that it operates in every tissue, but within each of those tissues, it governs the oscillation of hundreds or thousands of genes within each of those tissues, so-called output genes, which of course then distribute all over the animal, all over the animal's physiology. And in fact, as a consequence of these two principles, namely disperse among lots of tissues, plus the broad reach of its effect on gene expression within each of those tissues, it's estimated that at least 70% of genes in mammals, of the 22,000 genes that mammals have, is expressed rhythmically in at least one tissue. So in other words, this is incredible effect on all of gene expression, all of physiology. And that's the third reason why I received such a gracious introduction. We had a little bit to do with number 3. We had almost nothing to do with number 2, and number 1 was a lucky break, if I could call it that. I want to sort of segue to some recent experiments with a quote from Sidney Brenner, which I quite like, that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And I'll try and use that theme in order to, let's say, frame what I really want to say. So let me begin with a recent piece of work from my lab. And in fact, this is a long interest in gene expression and molecular biology is a long continuation of what I've done ever since I was a PhD student. This technique, which is in press, which we call BuTT-Seq, and the reason for that slightly racy moniker is because it really refers to the 3' end of transcripts, and the idea is, of course, to localize Pol2 on transcripts, which can be easily done by simply sequencing the 3' ends of nascent RNA, and then you know exactly where Pol2 is, because Pol2 holds on to nascent RNA, and the 3' end is the position that's closest to the actual position of Pol2. And the focus here, of course, is on circadian transcription regulation. And this is, in fact, a picture which illustrates what I just said, namely the fact that both elongating Pol2 and pause Pol2 are localized by the sequence at exactly the 3' end of nascent RNA. And like all of science, most techniques are an advance on something that already exists. And in fact, a collection of Pol2 techniques, especially nascent NeuroPSI, already existed, and this technique is similar, but it's just much faster and can be done with much less input RNA. And let me see if I've skipped a, I wonder where that is. Let's see, okay. So it has taken a 5 or 6 day protocol and turned it into a half-day protocol, and also allowed the use of much less material. And really the key to this is this enzyme from Alan Lambowitz, namely TGRT, which makes all of library prep much more efficient. And a result here, or a way to visualize the result, is to ask the question, how does a BuTT-Seq assay from head RNA isolated at different time points compare with RNA-Seq data? And the RNA-Seq data from 2 different data sets is shown here in blue and gray, and BuTT-Seq data, that is the actual Pol2 positions, is shown in red. And what I want you, and of course, these are time points across 2 days of light-dark cycle, and one gene, one classic circadian gene, the gene VRE is shown on the left, and the gene timeless is shown on the right. And what you are supposed to notice is that the transcriptional curve is completely superimposable on the RNA-Seq curve, the red line compared to the other 2 lines. In other words, the precise phase and amplitude of those curves are absolutely identical. And the implication of that is, of course, not only the transcriptional regulation is dominating the profile, but there's even another simple interpretation of this perfect superposition of the curves. That is that the mRNAs have to be extremely short-lived in order to have the amplitude and phase exactly superimposable. These are theoretical curves in which the mRNA has a half-an-hour half-life, 2-hour half-life, and 4-hour half-life. And you can see that as the half-life gets longer of a messenger RNA, the mRNA curves are both displaced to the right and their amplitude is reduced compared to the transcriptional curve. So the perfect superposition means that the RNA half-life has to be extremely short. And that makes intuitive sense, of course, because these molecules are turning over and you want everything to really be short-lived. So one slide here to sort of move ahead a bit and ask the question about the broad range of cycling RNAs. And this is a bit for the cognoscenti, but it makes 2 points. And this work is being written up. This is not published yet. Everything I've shown you so far can be found on bioarchives or published. The fact is that for those people who do this kind of thing, cycling RNAs to identify these hundreds or thousands of cycling RNAs is a formidable task, in part because the shapes are different, the algorithms for doing this are complicated, data from different labs are different for reasons which are not completely clear. And so, the question is how to proceed. And the student who's doing this, and I'll mention him in just a moment, he's done 2 things. First of all, he's really used machine learning to try to identify a set of genes which you believe, and then use those genes in order to extrapolate to a larger set. And in fact, plotting the genes turns out to be the best way to actually convince yourself that these curves are legitimate, right? So you can sort of see by eye the RNA-Seq curves in blue and green, and they're sort of credible, the 2 data sets. And in orange is, again, the BuTT-Seq data. And if you simply use these sort of new methods and then use plotting, as opposed to just an algorithm. So one of the conclusions we've come to is you really have to see it. If you see it, you believe it. And if the computer tells you it's there or it's not, so many genes which don't make algorithmic cutoffs are totally convincing by this criterion. And so it's just simply that the different algorithms for defining an oscillation are imperfect. So the conclusion from all this is really that transcriptional oscillations also governs the large number of genes which are oscillating in different places. And I think it's to our mind, doubtful whether there's really major contributions from post-transcriptional regulation in the various places that have been looked at. Because when we've really ground down into this, it looks like transcriptional regulation is the major, is a predominant contributor to this oscillation. So this method BuTT-Seq was really designed in order so that we could assay small amounts of material. And the reason for being interested in small amounts of material in my lab is because of the 75 pairs of neurons bilaterally placed in the two sides of the fly brain, these so-called clock neurons, which govern the behavioral rhythms that we see of the fruit fly. Activity in the morning, activity in the evening, a siesta in the middle of the day, and then nighttime sleep. And my lab got interested in this about 20 years ago, almost exactly at the same time as Francois's lab here. And France got interested in this. And so we have been interested in this problem for a long time, which has dragged me or made me into what Woody Allen described as a neo-mock pseudo-faux, a neuroscientist. And so it's with that second disclaimer that let me move into the neuroscience part of my presentation. And so perhaps I neglected to mention that most of the biochemistry that has been done is on fly heads. And so, of course, we're interested in these clock neurons as individual entities. And that poses a significant challenge because the amount of material is very small. And so this also, these 75 pairs of neurons, of course, and the behavioral program I mentioned to you, of course, link to this more general problem, and you'll hear about this from Gero, that the circadian clock, as well as homeostatic principles, govern the sleep-wake cycle. And of course, this fly behavior I showed you is really the flies equivalent of this mammalian sleep-wake cycle. So with an interest in these very small amount of neurons, we've used intact, that is the purification of nuclei from tiny amounts of material with GFP on the cell surface of the nuclei in order to sort of do some simple biochemistry. And I'm just going to show you two soupçons of data here to give you an indication. First of all, if you look at cycling transcripts from these circadian neurons, it turns out about half of them are completely novel, as shown in red. That is, when you look in fly heads, there's no evidence of oscillation. And this, of course, leads to interesting principles of gene discovery, function, and mechanism. That is the ability to really look at time points in these very small numbers of neurons. And of course, distinctions between the different kinds of neurons as well. And secondly, to link to a principle that you heard in the first talk yesterday, you can also take these neurons and you can do ATAC-seq, that is, look at open chromatin in these purified neurons shown in the green line on the next to the bottom. I'm not quite sure I can figure out how to use the pointer here, and then using BuTT-Seq on these purified neurons, you can find so-called enhancer RNAs, that is short transcripts, which are coincident with the BuTT-Seq peaks. And so those are putative enhancers that are much more prominent in the purified clock neurons than they are in heads. And they coincide with transcription factor binding sites as defined by ATAC-seq only in the clock 856 data. And so we can use this technique to really hone in on principles, molecular biology principles that are specific to the neurons. Okay, so the person who's pioneered this molecular biology effort in my lab is a student, Albert Yu. He's what is known in the United States as an ABC. He's an American-born Chinese, but but Albert, and this is a human tangent, a human aside. Albert is the son of this guy, Chang, who was my first PhD student from China in the mid 80s. And he did some of the most important work on circadian rhythms in collaboration with me and Jeff. And so Chang and his wife, Ji Ying, stayed in the United States for a very long time. Albert was born a few years after he left my lab. And so Albert is a second generation PhD student, if you can imagine, if you can imagine such a thing. So that's so unusual. I thought I couldn't avoid saying that. So this is the title of my seminar, of course. Can we exploit this circadian system to learn some general principles about the brain that might be interesting? And so this part of the seminar is done by a postdoc who's on his way back to a faculty job in China, Dylan Ma. And the question at the outset is really, how heterogeneous are these 75 pairs of circadian neurons? How many cell types are there here? And you can see that the traditional identification of these cells put them into 7 or 8 groups based on anatomical location, and there's been immunohistochemistry done. So we know from traditional means, let's say anatomical identification one way or another, that there are 7, 8, 10 groups. And so the question is, from single cell sequencing, how many groups do we really find, and what might that tell us? And so the way this is done is to take time points of fly heads, because we were interested in oscillation, as I indicated, oscillating molecules in these neurons. And we took time points around the clock, both in light dark cycle and in constant darkness, a paradigm of interest to circadian biologists. And we had, in the end, a very deep coverage of the cells. In other words, about 10 copies of each cell at each time point. And this will become relevant for some of the general points I want to make. And so we did this, and to make a long story short, we found in this subset of the circadian neurons, about half or 60% of those 75 cells, that's the subset we were working with. We found about 17 different clusters, 17 cell groups, or about 2 times more than the number of anatomical groups that had been originally identified. And the important principle here is that assigns about 2.5 cells per brain, per hemibrain, to each cell group. So that's the number of copies of each cell type that the fly contains based on this kind of analysis. And so I want to impress you with the fact that that's a very small number of cells for each cell group. And so the second principle that emerged from this work is that there is prominent expression, cell type specific expression of cell surface molecules. And this is the adult, fully developed, 10-day-old, mature fruit fly. So the reason that distinction is relevant, of course, is because these molecules, these cell surface molecules, are very important for wiring of the nervous system. And they've been studied in development in the fly, most notably, I think, in the visual system by Larry Zabersky and Kai Zinn. And the code by which these cell surface molecules interact with partners has been worked out. And of course, this is the way that a developing neuron, a growth cone, developing axon, finds guideposts and decides to turn because of these kinds of interactions, and ends up, at the right place, the retinal tactile connections, et cetera, same principles that have emerged from mammals as well. And so what was unprecedented and intriguing, I must say that an old yeast competitor colleague of mine way back when I realized one day, listening to her that she used the word intriguing in a very positive way when she referred to her own results, and in a very pejorative way when she referred to somebody else's results. In any case, I'm using it in a positive way, of course. So the conclusions, the summary from this sort of first part, which was published a year or two ago, is that there's dramatic heterogeneity of the clock neurons. Am I doing something wrong? It's not. You trying to find my pointer? It's that one there. The small thing there. Okay. Okay. Ça vaut pas la peine. C'est tout petit. Okay. So there's dramatic heterogeneity, about 2 to 3 cells per cell type. There's adult expression of these cell surface molecules. And I haven't shown you, in the interest of time, I haven't shown you the experimental result, which shows that if you eliminate one of these molecules only in the adult, you get a behavioral phenotype. So in other words, these molecules and their expression in the adult are important for sleep and wake, right? And of course, the suggestion is that adult transcriptomic diversity underlies anatomic diversity, and I'll have more to say about that in a couple of minutes. So for those of you who don't care about circadian neurons, which is almost everybody here, you could ask the question, well, are these general principles, or are they just restricted to circadian neurons? A question we asked ourselves. And so we basically repeated the entire exercise I just described on dopaminergic neurons. And so we purified about half of the dopaminergic neurons in the fly brain, which are about twice as numerous as the number of circadian neurons. So, you know, the factor 2, a couple of hundred dopaminergic neurons in the fly brain. And we actually did this in a way in which we made a fly which expressed GFP both in the dopaminergic neurons and in the clock neurons, as indicated by the 2 colors. And then we did the analysis, the purification, the single cell seq all together. And we could, of course, use our previous data on the circadian neurons as a benchmark in which to compare the data here, and then bootstrap from those validated data, if you will, to the dopaminergic neurons. And so when we did that, we ended up with a lot of clusters, and we could identify which cluster came from where because of the marker genes that are characteristic. Only the dopaminergic neurons make tyrosine hydroxylase, make the dopamine transporter, et cetera, and the clock neurons express the clock genes, and they're mutually exclusive. So it's easy to parse the clusters into one or the other. And the important conclusion here is that the number of dopaminergic cell types was exactly the same, 2.5 cells per cell type, as we got from the circadian neurons, suggesting, of course, that that conclusion is more general than just being restricted to the circadian neurons. And importantly here, there are 2 very high-profile papers, maybe 3, in fact, that were published in the last 3 years or so, in which the entire fly brain or heads were characterized from the cell type point of view and asked how many cell types do we have, et cetera, et cetera. And the point is that there was nothing apparent in those data that gave rise to the number of different cell types that we identified, both for the clock neurons and for the dopaminergic neurons. And it's almost certain, I would bet a lot of money, not my children's lives, but a lot of money, that explanation is correct, that they simply didn't assay enough cells, and consequently, you're missing the rich diversity intrinsic in each of these cell types. And so the question then is, where do we go from here? And so the cells are clustered. This sort of clustering or identification of cell type is done by identifying a set of highly variable genes, which can be used to identify cell type. And so there's 338 highly variable genes. There are marker genes that are distinct for each of those different clusters, for each of those cell types. And then you can ask the question, well, what are the most common Go terms? What are the most common molecules, which are the highly variable genes, which define cell type? And here they are shown on the right, transcription, and the number of them is shown out of 338. There's transcription factors, cell adhesion molecules, which are identical to cell surface molecules. So it's a synonym. GPCRs, neuropeptides, et cetera, et cetera. And you'll notice that three of those four molecules, 1, 2, 3, 4, are molecules which have to do with cell neuron communication, right? Transcription factors in 3 categories of cell communication. And if we take the cell surface molecules, in clock neurons and dopaminergic neurons, those are the heat maps of those 2 categories of molecules. And on the right is a forced embedded layout that asks the question, to what extent can cell surface molecules alone, only these 56 cell adhesion molecules or cell surface molecules, to what extent can they identify cell type? And the answer is pretty good. In fact, very well, because there's each cluster is distinct, and there are very few links between the clusters, indicating that kind of protein, that kind of gene alone can identify cell type. And if you compare that to transcription factors, and there are the identical number of transcription factors in cell surface molecules, you can see that transcription factors do much less well in identifying cell type, especially for the clock neurons shown in gray. So they do better for dopaminergic neurons, but you can see even the dopaminergic neurons are more linked with transcription factors than they are with cell surface molecules. And so the champion really here is GPCRs, G protein coupled receptors, because they also do extremely well in defining cell type all by themselves. And of course, there's only 21 of these compared to 56. So if you think of specific activity, the ability of a number of molecules, genes, to be able to define cell type, GPCRs are the champion, because there's fewer of these, and they do an excellent job alone in defining cell type. And so, for the people who are not familiar with this, GPCRs bind ligands. From a neuroscience point of view, most interestingly, neuropeptides, perhaps. And so the interesting thing here is, of course, that anatomy is being defined, especially in the connectomics of the fly, based on anatomical principles which identify synapses. But neuropeptide GPCR interactions don't have any defined synaptic components that make it easy to identify, for instance, in the electron microscope. And the implication is, of course, that circuitry is even more complex than defined by anatomy based on GPCR neuropeptide interaction. So the second summary is that there's dramatic transcriptomic heterogeneity of dopaminergic as well as clock neurons. There's result-specific expression of neuronal communication molecules, the best definers of cell type. And the speculation, of course, is that transcriptomic diversity underlies anatomic diversity. And so the sort of 2 subsequent conclusions, important and general role of communication molecules in adult neurons, probably for maintenance or even plasticity of circuits, adaptation. And the last point I already made, that the GPCR data suggests that neuronal circuitry diversity may be even more complex. So I'm going to, in the last 3 minutes or so, 5 minutes at most, give you a little unpublished update and simply say that we also wanted to address the missing part of the circadian neurons, the 75 neurons that our previous work had not done. And you can see here in the driver that we use to identify the circadian neurons, shown in GFP, and timeless circadian neuron markers shown in red in the middle, that there's a bunch of clock neurons shown with the arrow on the right, which are red but not green. In other words, they are not identified in any of the analysis we've done. And these are the so-called enigmatic DN3s, about which there is extremely little work done in the fly circadian behavior world. But they represent about almost half of this total 75 neurons, maybe 35 or so of the 75 are those DN3s. So we went trying to analyze these data to, in other words, to try to complete our circadian neuron story. And the important point here is, I'm not going to tell you much about that work, but the important point is that we expanded by a factor of 5 to 10 the number of cells that we examined in the course of this most recent unpublished study. And of course, I've already hinted that the number of cells, getting the number of cells larger and larger, gives you more insight into some of these principles. And so this most recent study has looking at many more cells, and this is now back to Sydney, and I will segue from techniques to discoveries. And of course, by discoveries here, the discoveries is a parole un peu gonflée, but it's this a finding, an unexpected finding, a band on a gel, something you stumble across and say, well, what is that, right? That's a discovery in this modest sense of the word. And so I want to return in this context of examining more cells to the original 75 neuron picture and focus on these so-called LPN neurons, of which there are 3 by anatomical definition. And everyone, including our transcriptomic work, defined 3 cells there. So it's very simple, very straightforward. There's always 3 cells. And when we examined 10 times as many neurons as we had before, damn it. Okay. It turned out, I'm missing the slide, or it didn't show up on the right. It turned out that the work divided these 3 cells, this one cluster, into 2 clusters, one with 2 cells and one with 1 cell. So in other words, the larger number of cells actually gave us an increased heterogeneity, increased number of clusters. And it turned out, this really isn't showing on the right. Damn it. Gero, you insist on using your own computer, because this is my presentation in their computer. They're going to connect you to the internet. I warn you. Okay. So in any case, you can't see the pattern. You can't see the pattern on the right. But suffice it to say, there are 2 clusters. One has 2 cells. One has 1 cell. And it turns out that cell surface molecules are distinct in these 2 clusters. The cell surface molecules that I've been talking about beautifully distinguish the 2 clusters. And maybe Gero will be interested that DOP2R is exclusively in one cluster as opposed to the other. And so it turns out that when we got these data, we turned to the Janelia connectomics, and it turned out that these 3 LPNs have been defined in Janelia as 2 cells with the connection pattern in green on the left, and 1 cell with a connection pattern in purple on the right. And here are the 2 patterns overlaid, and you'll notice this purple branch is distinct to this 1 cell and not present in these 2 cells, right? So in other words, with the split that we observed with many more cells and the differences in molecular composition, the transcriptomics turned out to have a perfect parallel in the connectomics. And of course, the speculation is that, like DNA makes RNA, makes protein, right? That the gene expression in these neurons, the adult gene expression even, underlies the anatomy and connectomics. And of course, that produces behavioral distinction. And of course, we're interested in who these connect to and exactly, what the difference is. So I want to finish with one last piece of speculation. And that is that this remarkable heterogeneity in the fly brain suggests that there are, an incredible number of cell types, which of course implies a very large number of circuits that can be made, even leaving aside plasticity, adult plasticity. And so I think this can explain why this tiny organism with this tiny brain has such sophisticated behavior, simply because it can build this dramatic number of circuits with this very heterogeneous collection of cell types. And so I'm right on time, and I thank you for your attention, and I'd be happy to take any questions.